So we started to talk about the coordination complexes in living systems. So I mentioned from the previous video porphyrins, which can be found, porphyrin rings can be found in the hemoglobin, but there is others as well, hemocyanin, hemoerythrin compounds, and we can find the porphyrin rings in the photosynthesis in chlorophyll, which involve electron transfer, vitamin B12, and so on. So here I showed you porphyrins in hemes, and we talked about these metalloproteins. So from the name is metal, there is a metal and there is a protein around it. And they are giant biomolecules. So in biology, biomolecules, I mean it's in biology, which contain metal atoms. And we talked about the lungs and how transfer oxygens from the lungs to the blood, to the human cells and or living cells, and then come back to release them and bring carbon dioxide to the lungs again to get rid of them. Vitamin B12 cobalamin, we discussed this before. Very important to production of blood cells, red blood cells, to have normal function of brain, nervous system, synthesis of regulation of DNA, fatty acids, and only bacteria, archaea, have the enzymes required to synthesize this vitamin. This was the chlorophyll we talked about. There is a magnesium in the metal center. In the center of the molecule, there are four bonds from four nitrogens. And this is as well around the magnesium is the porphyrin ring, but always we have, okay, the difference between the hemoglobin, chlorophyll, others is what functional groups are linked to the porphyrin ring. So that's the difference. So now we come to the hydrolytic enzymes. This was a very like it was my research specialty, and we the research was about PhD research was about the polypeptides, macro molecules. So there is a micro and there's a macro. So macro molecules, they are big molecules from or formed from the alpha amino acids, linked by peptide bonds, which like a cage, and formed by the elimination of water. So you can see here to the right side, the structure I just drawn. There is a carbon, oxygen, nitrogen, hydrogen. This is the main core of the polypeptide and amino acids. So these, whatever have outside the cage, will be different functional groups, and they can give me a different amino acid. But the main amino acid is the CONH amino acid. So the amine is nitrogen. Acid came from the carboxyl, C double bond O. Okay, so two important hydrolytic enzymes are carboxypeptidases, A, and thermolysin. Carboxypeptidase and thermolysins. So peptide hydrolysis is difficult despite being thermodynamically favorable in aqueous solution. Okay, so hydrolysis is difficult. In order to catalyze this reaction, we need an enzyme. So the enzyme must facilitate the nucleophilic attack on the peptide carbonyl group by a nucleophile. Second is stabilize the tetrahedral intermediate transition state that is generating following the first step. Stabilize the amide nitrogen atom to make it suitable leaving group so the tetrahedral intermediate can collapse upon C-end bond cleavage. So cleavage means breakdown. Zinc metal ion might play an important role in any of these processes. Over years, peptidases mechanisms being suggested to understand the functions of the active site of the zinc ion. So now you can see here to the right side, we have zinc metal bonded to two nitrogen and two oxygens. And there is a water group. And these two oxygens here, I can show you. These two oxygens from the glutamic acid. Here, the nitrogen bonded from the histidine imidazole group. And here's the imidazole from the another histidine. There is a water molecule bonded to the zinc. Okay, so this is the hydrolytic enzyme, and the zinc is important for the core of the active site. In all these compounds, in all their compounds, cations are surrounded by anions or neutral molecules. Groups immediately surrounding a cation are named ligands, as we know, and the branch of inorganic chemistry concerned with combined behavior, cations and their ligands is coordination chemistry. Metal ions are usually covalently bound. Okay, so now we have a carbonyl. This is the carbonyl CO, C double bond O to the metal center. We have bi or chelate, carboxylate. We have monodent, one bond from the oxygen to the metal. We have monodentate bridging. This is monodentate bridging between the two metals. We have bidentate bridging, carboxylate. We have imidazolyl. This is imidazole ring, so it's bonded to the metal. There is amino group, NR3, to the metal. And there is a syn amino and anti-syn amino group to the metal. So metal ions are usually covalently bound to the polypeptide backbone by endogenous ligands provided by amino acid side chains. The scheme shows the most common biological ligands that proteins utilize to coordinate to the metal ions. So these are, we studied them in details during my course. Now, if we come here, we can do some animation. And if we do, sorry. So if we play that, sorry about that. came all in bulk. So here what we have is carboxypeptidase A, and here we add some animation. Oh my God. So it's all section came in at once. 3 obtained forms of bovine or cow pancreatic carboxypeptidases. So carboxypeptidase A been obtained from the cow bovine pancreatic or pancreas, designed as a alpha. So here it talks about the carboxypeptidase, which it's been the structure uh, being talked about in details, and how is coordinated to the zinc, where is the zinc, and which groups around the zinc bonded. And there is a dentate, there is, in the presence of the substrate or inhibitor, the glutaric 72 carboxylate group becomes coordinated to unidentate fashion. And this is talking about the structure and how everything is bonded. And I don't, yeah, it's in the figure. So the metal ions can be substituted because of the zinc 2 is magnetically and spectroscopically silent because of the D10. Cobalt 2 derivatives can substitute zinc 2 and result is more active than native enzyme in hydrolyzing peptides. So manganese 2, cadmium 2 show less activity. If we replace zinc by cobalt 2, by others, so there will be some difference in the activity. You can see here the zinc. This is in the center of the molecule, structure of carboxypeptidase A from bovine pancreas and its zinc-based active site. So here in the active site, this is the active site where you can find the metal, zinc, and the surrounding group. So this is the active site. This is the heart of the molecule, of the supermolecule. So the rest will be a protein. These are protein sequences or bonds, and it's a huge molecule. Carboxypeptidase mechanism is being studied in details, which is the tetrapeptides with the cation values 20 to 500 per second, depending on. So everything here you can see ligand and metal reaction, how there is a water with the metal losing one hydrogen, so it will be hydroxy metal. If gain hydrogen, proton hydrogen will come back as a metal ion and water, or an aqueous solution. So every pKa been studied as well. And here you can see the zinc 2 histidine groups, carboxy from the glutamic acid water molecule in different coordination. So everything here is being studied. I'm just quoting from my thesis. So mechanism emerged as there is a terminal amino acid reside of a polypeptide chain binds to the active site, orienting itself, and then continue. Carbonyl group of the cis peptide bond being hydrolyzed and interact with the arginase, a group increasing its Lewis acidity, and the rest of the mechanism being explained in details. So here is the drawing, what's happening, how is you introduce substrates and then changing coordination, changing structures, introduce water and substrate of phenyl group, and then you can find the structure from the beginning. So this is the mechanism for peptide bond hydrolysis by carboxypeptidase. So the carboxypeptidase is the enzyme which is increase the speed of the reaction for the hydrolysis. So, and everything here is being studied. Lyase, carbonic anhydrase. So lyase shows zinc 2 to the coordination, and there are other functional groups. Kinetic, the motion of the carbonic anhydrase reaction is a pH dependent. So being faster at high pH under the control of a group having pKa value at 7, this reaction like that for carboxypeptidase equation here mentioned, and the rest you can study that. I'm just giving like this is the basic of what we are coming to discuss. Others, and these been obtained from the journals, scientific journals. This is the carbonic anhydrase. Carbonic anhydrase. Again, there is a zinc, and there are different groups, functional groups in the heart of this enzyme, carbonic anhydrase, and the geometry of zinc active site. This is how introducing carbon dioxide, changing the structure, and then introducing water, releasing carbonic acid to come back and lose one proton to return to its normal condition. So this is the carbonic anhydrase mechanism, a scheme for hydration of carbonic dioxide. Now, this is my research, depend on this enzyme, urease. So urea is formed in large quantities as a product of the catabolism of nitrogen-containing compounds. Each human produces about 10 kg of urea per year. Sorry. Urea slowly decomposes with a half-life of 3.6 years on its own without enzyme. Biological catalyst for the hydraulic decomposition of the urea is the enzyme urease. So urease catalyze the hydrolysis of urea. And what is happening this? Urease catalyzes the hydrolysis of urea in plants, algae, fungi, several microorganisms to produce ammonium and carbamate. So the health and environmental impact of urease activity is enormous. And control of the rate of urea hydrolysis by using urease inhibitors will lead to enhanced efficiency of urea nitrogen uptake by plants and to improve therapeutic strategies for treatment of infection by ureolytic bacteria. So you can see here, you have the urea, carbon double bond O, NH2, NH2. When you introduce water with urease enzyme, what you do, you replaced the NH2 by oxygen and leaving ammonium out. So once you have this intermediate, more water will decompose this to NH3 and carbamate. So we call this a carbamate. That's the mechanism of urease. Crystallographic studies of the native enzyme. This has been obtained from the journals, and we used it to explain the prepared, or we mimic the active site of the enzyme, and which shows the similarity with the enzyme itself in biology. So the X-ray crystal structure of Klebsiella aerogenes urease provided the first detailed three-dimensional structural model of the active sites. In native Klebsiella aerogenes urease, two well-ordered nickel atoms are held at the distance of 3.6 angstrom by the bridging carboxylated groups of the carbamylated lysine residue. So everything here is the crystallographic studies of the native enzyme and the functional groups with the distances and naming of the groups with the pKa and dissociation of the acid. And here, this is the active site of the urease, which contain two nickel sites, bridging water, a bridging carboxylate, and there is a water-water molecule. Let's see, check each metal center. So every metal center is coordinated to 3 oxygen and 2 nitrogens. Here, the center is penta-coordinated. Here is octa or 6 coordination here of the second center, metal center. So 5 by 6. This is the representation of the active site of the Klebsiella aerogenes urease. Proposed mechanism for the hydrolysis of urea by urease. Here we propose this. These factors include the followings: urea, oxygen coordinate, and this is in details how is happening the mechanism. And these are this is my designs of mechanism of the hydrolysis of urea by urease enzyme. Nucleotide activation. Several zinc enzymes are involved in nucleic acid metabolism. Okay, so transfer in DNA, RNA, polymerase, and reverse and the other essential. All enzymes on the nucleotide, such as ATP, require magnesium 2, Mn plus 2 for the activity, and these as well being talked in details. And now I'm focusing on the dinuclear nickel active site. Where is it gone? I don't know what's happened here. So I'm talking about the active site, and many diagonal nickel 2 system received considerable attention recent years. The precise details of urea binding within the enzyme active sites Its catalytic, this is important, a catalytic conversion was first suggested by scientists Zerner, Lippard, Benini, and Karplus et al. Synthesis and investigation of small dimeric nickel-2 coordination compounds in order to elucidate possible coordination modes of urea to probe their relevance to the metal-assisted hydrolytic degradation of this important substrate. So then I talked about the mechanism of the metalloenzymes, the main subject of interest. And we're here to provide the di-nickel active sites of urease simulated by synthesis of series of model nickel-2 coordination compounds. So urease models should have, here is important. So urease models should have oxygen and nitrogen donors in the first coordination sphere using carboxylates, water, hydroxide, phenoxide, alkoxide, and sometimes an ion such as perchlorate, CF3SO3 minus as oxygen donor ligand, and nitrogen ligands such as ammonia, tetramethylene diamine, tetramethyl ethylene diamine, bipyridine, pyridine, thiocyanate ion, imidazole, or benzimidazole. So the second is dinuclear structure of the metal-metal distance should be less than 4 angstrom to mimic the active site of the urease enzyme. Terminal nitrogen ligands to exhibit histidine-like ligation. We need to have mono or bidentate carboxylate as a bridging moieties. Metal-bound hydroxy aqua group. But there is no ideal design compound so far synthesized because of the difference in coordination, environmental, and charge between model compounds and the enzyme. So these models being prepared, okay. And as you see here, dinickel, dizinc, and we changed the groups around, and these compounds being prepared in our labs in University College Dublin for years, and crystal structures been obtained, and metallo enzymes, metal ion centers could be nickel, cobalt, zinc, manganese, iron, anyone. And then many, even clusters being prepared, cobalt 9. You have, I prepared the manganese arginase model, ornithine model, groups of TMN, tetramethylethylene diamine, being used, should be one word. Imidazole, IM, bridging, mu, arginine, ornithine, carboxylate, methyl, ethyl, and so on. So here, where I obtained the urea with water in the presence of urease enzyme, so it's been dehydrated or hydrolysis of the urea to form a group ready to coordinate, and there is ammonium produced as well. This is exactly what I took it from a scientific book. So atom and group transfer chemistry, we of course we studied the group transfer and how we can use the bioinorganic chemistry of dioxygen binding was well described, these principles and the rest. Dioxygen transport, non-enzymatic role, reversible dioxygen binding. This as well, it was in my thesis, and the dioxygen carries carriers of hemoglobin in vertebrates, many in invertebrates as well, hemocyanin in arthropods, mollusks, hemerythrin, non-iron functional core in 4 phyla, marine species. And there's a table here to summarize the 3. So I talked about myoglobin, hemoglobin, and others we studied. This is very good for bioinorganic chemistry. So there are some properties of protein oxygen carriers and being explained in this table. So how a metal oxygen, color oxygenated in the red, violet, pink, blue, and these hemoglobin, hemerythrin, hemocyanin, and everyone has a different characteristic. Values here depend on the molecular weight, number of subunits, oxidation state for each, and there is iron in hemoglobin, iron 2 in hemerythrin, but copper 1 in hemocyanin. So I talked about hemoglobin and myoglobin in details. So the structure of each myoglobin being determined in all 3 deoxy, oxy, and metallo forms. And there is how we can find it and how the binding of the dioxygen occurred, and the bond angle, distance as well. All there is been published in papers before us. Structure of myoglobin, this is it. This is the active site, and this is the protein. There is iron, porphyrin ring, but that's the structure of myoglobin. Now, physiological function of the hemoglobin, myoglobin, which oxygen gas binds to the hemoglobin in lungs and is transferred to myoglobin in tissues or to fetal baby, hemoglobin in the uterus of pregnant mammals. So everything being explained here. You can see the coordination of this. High spin iron 2, there are 6 electrons surrounded or bonded to the, or have the iron 2. It has, you remember, 4s2, 3d6. So losing 2 electrons, leaving 6 electrons. And as we said here, you have it as 6 electrons in this square planar. And here, when the oxygen is bound to the iron from the top, so what will happen? So it comes from the dx square y square, and then you can see losing 1 electron to the oxygen to bind, and the dioxygen will come as a ligand. So one oxygen needs an electron, take it from the iron, and then the iron become iron 3, which will be, again, it's a low spin with 5 electrons. Model chemistry, it's easy to synthesize iron 2 porphyrin complex with physical structural properties similar to the metal centers in deoxy hemoglobin and myoglobin. So deoxy, it means without oxygen, and oxy, it means with oxygen. So there is some examples here. So the iron 2p plus oxygen is bonded. That's oxidation. And then when there is another iron combined with this iron 3, when iron 2 comes, iron 3, so it will be bridging oxygen, changing the oxidation for both to be 3. And then the formula will become iron 4. And this iron 4 will combine with another iron 2 to become 3 by 3. Hemerythrin is here deoxyhemerythrin or oxyhemerythrin. So here you have the deoxy one with by iron center oxidation number 2, but with the oxyhemerythrin, losing 1 electron from each iron will become oxidation number 3 by 3. And this, we call it oxyhemerythrin. Here is deoxy. So deoxy without oxygen, oxygen will come and bind to one metal center, and then oxidation number is changed. So now you can see here 2 iron center. There is a bridging oxygen. There is carboxylated groups too. And we have other bondings, other bonds. And each center is how many? 2, 4, 6, 2, 4, 5, 6. So it's octahedral. And this is the center of the hemerythrin. Hemerythrin model compounds. So we introduce a model compound similar to the hemerythrin. So the iron here is a green-brown molecule with HbF4 forming orange molecule. And then you can come back with using the N-ethyl tri group to come back with the same molecule. So this is it. This is a model compound for hemerythrin. Hemocyanin, what's in hemocyanin is copper. Again, we studied this or been studied before by X-ray crystal structure of hemocyanin obtained from the spiny lobster arthropod, and dinuclear copper center has been shown. And then, oh my God, look at this. So this has been terrible. Just I wanted to do bonds between them. So there is a 2 copper Cu centers. And the bonds here, I just wanted to put them together. And you can see there is a bridging oxygen and 2 copper center, copper 2. So this is the main or the active site of the hemocyanin. You can see here the in reality protein outside and 2 copper center in the middle or the active site. You have 2 coppers, nitrogens, carbons. So we can see here the groups of surrounding the active site of the hemocyanin. And then there was a study of the copper centers, how they are, and then schematic views of the structure, deoxyhemocyanin in the top and oxyhemocyanin at the lower bottom, showing the binding of dioxygen eta-2 peroxide. This is the oxyhemocyanin models. Here is the model, here's the active site. Protective metalloenzymes. You have here the copper zinc superoxide dismutase enzyme. And this as well was the approach we used to come to reach to our models, whereas copper is there and zinc. And this is the structure of copper zinc superoxide dismutase and its catalytic dimetallic core. So there is a zinc and copper in the metal center. Enzyme and reaction of the cytochrome C. This was my project for the bachelor degree, 4th year. This was my project. Prepared cytochrome C from the liver of the bovine. And then here you have the horseradish, spinach peroxide, and the others reaction of a heme containing peroxides and catalases. So you can see the porphyrin ring is being distorted. If this is the iron, and there is imidazole ring here from the histidine, here from the thiocysteine and tyrosine when it's bonded to the iron. So here you have the horseradish peroxidase. Here is the chloroperoxidase and is the catalase. Dioxygen radical generating systems as well. So you have enzyme, and we have here RR enzyme catalyzes the conversion of ribo to deoxyribonucleotide to the step of biosynthesis of DNA. So this is as well structure we depend on. Coenzymes B12 dependent reaction. That's been discovered in 1926. B12 reaction catalyzes by coenzyme B12 dependent enzyme. You see the reaction, and here is the ribonucleotide reductase. And there was an example here from the book to study. And then I reach here to stop, to continue. All of this was the introduction to bioinorganic chemistry, bioinorganic. So we depend on the biology to introduce bioinorganic chemistry in the lab.